And definition of evolution is descent with modification. So the genes get passed on, but with modification, and it happens over generations and generations, and it works on populations. It doesn't work on individuals. So that's evolution. Let me see. Can you guys define natural selection? Think about the pepper moth example.
Survival of the fittest.
Survival of the fittest, and the genes that are favorable, alleles that are favorable, they get selected. Mutations that are beneficial, they get selected. What is the difference between natural selection and artificial selection?
Artificial involves a human selecting the genes that are favorable.
Yeah, so maybe like Chris' dog, maybe someone picked those genes for his dog, so humans did do that. So there's human interference. For natural selection, it's happening naturally. So I might give you an example, and I'll ask you if this is artificial selection or natural selection. Then there are two vocabulary terms, microevolution and macroevolution. Please write the definition for that. You will see it on the exam. Microevolution and macroevolution. It is on your vocabulary. Are you ready for this chapter list? And then if you want to calculate allele frequency, this is the formula, number of copies of an allele and total number of allele for the same gene in the population. We said if red is 160, total, maybe little r is for pink is total 200, you're gonna do 160 over 200. So that's how you apply that. I'm probably not gonna have you do any calculations, so just remember the formula. And then definition of gene pool. Definition of gene pool is the entire collection of genes and alleles. So entire collection of genes and alleles. So I might say, what is a gene pool? Then you have to tell me. Who is responsible for the theory of evolution?
Darwin.
Charles Darwin. What species did he work on? Which place?
Finch in the Galapagos.
Finches in the Galapagos. And then difference between absolute dating and relative dating. That's probably next chapter, but I just want to know if you guys remember that. Absolute versus relative dating.
I might be wrong, but doesn't relative, it's when you use multiple rocks in the area to date the location. Absolute is the specific date.
Yeah, so absolute is, let's say we're looking at this rock layers. Top ones are more recent compared to the older ones. There's no number we are saying, oh, this is this many years older. There's no number we're applying. We're just saying they're more old than the top ones. Whereas absolute dating is when they're using those radioactive isotopes. So that's carbon dating. They're using chemistry for it. This is a place Darwin worked at, Galapagos Islands. Here is where he got the theory of evolution, and the name I want you to remember is Charles Darwin for this. I might ask you that. And he worked on the finch species. Okay, then this is showing artificial selection. So I told you this is wild mustard. You probably see it on the side of the road or if you go for a hike. And humans, they interfered with the genes. They started selecting for certain genes and mixing them. And now we have broccoli, Brussels sprouts, cauliflower, kale, cabbage. These vegetables, they do not exist in nature. So this is coming from human interference. Darwin did that with pigeons. Mike, one more person, I can't remember his name, is it Mike? He also is working with the pigeon coop and he's selecting for the genes.
Mike Tyson.
Mike Tyson, yeah. So he selects for the genes that he likes, and that's artificial selection. Okay, this is where some of you guys get confused. So natural selection, how it works. There has to be variation in the population. So something has to get selected for. If it's just one gene, there's nothing that can get selected for or selected against. And then the conditions has to be favorable for one gene. And then those genes has to be passed on. That's heredity. And over time, the favorable genes are going to increase in number. That's natural selection. And then some of the observations for that is genetic variation. So we just said that it has to be variation. Limited resources, that's going to create competition. So that's a type of environmental stress. So limited resources going to create competition. Also, if there's overproduction offspring, that's also creating competition because more are born than maybe the environment can support. And then that's going to create struggle for existence. These are the interference. There's a table in your book, so try to do a flashcard for this. Struggle for existence. So competition creates struggle for existence, and that's going to give unequal reproductive success. Means the individuals that are able to get the resources, they're going to pass on their genes. They're going to be able to reproduce, and their genes going to be getting passed on. So that's a natural selection. And the ones who cannot get the resources, they cannot live long enough to reproduce. Their genes do not get passed on. Therefore, they're not getting selected by the natural selection, or their genes are not getting selected for. And then over time, that's going to lead to descent with modification. In other words, evolution. So over many generations, keyword, many generations, doesn't happen overnight. Natural selection can change the characteristics of populations, not an individual, and give rise to new species. The term for new species we learned later was speciation. And then some of the things that natural selection leads to is being able to camouflage, because that's going to help them live better, live longer, and be able to reproduce. So natural selection is molding evolution. So it's kind of directing evolution depending on what's getting selected for. But then I asked you this last time too. Do you guys think it's only natural selection just leading to evolution?
No.
No, so there were a few other things. Do you remember what those were, Justin?
I know like one of them would be like environmental changes.
Environmental changes, mutations. So we'll discuss that next chapter. I think it's in next chapter. So, or maybe in this chapter, I don't know. I think it's coming up. So this is showing again variation, overproduction of the offspring, and it's all that is leading to competition, then struggle for existence, then some genes get selected for, and then they get passed on. So this is another antibiotic bacteria example for the same thing, another one for the peppered moth and light moth example. And then the term coevolution. I showed you the video about the bat and this flower that gets pollinated. You guys remember that? I think it was TED Talk. So that one is also an example of coevolution. So only that bat can pollinate those flowers. They're dependent on each other. If the flower wasn't around, the bat wouldn't get the nectar. And if the bat wasn't around, the flower wouldn't get pollinated. So that's coevolution. Here it's happening with the orchid and the wasp. They are evolving together. They wouldn't be able to evolve if one of them didn't exist. So here, so it mimics the female wasp or male wasp, the way wasp looks, and it comes and hits the head and grabs some pollen, and that's how it's pollinating. And then fitness, survival of the fitness is, so the term fitness is talking about reproductive success. It's not talking about physical fitness. Do you guys remember how we compare if there is a change happening in alleles? So if there's no change, we have a term for it.
Harvey-Weinberg equilibrium.
Hardy-Weinberg equilibrium. That means it's at equilibrium, nothing is changing. So allele frequencies always change. And if there's no change in allele frequencies, that means there's no evolution. And no change means over generations. If there's no change over generations. And we call that Hardy-Weinberg. So the definition for that would be when the allele frequencies do not change. They are equilibrium. So I will say, when can you apply Hardy-Weinberg? You're gonna say when the allele frequencies do not change between generations. But that's rarely the case. It's always changing. So natural selection does happen. So if it's gonna follow Hardy-Weinberg only when there's no mutations, and you can't really guarantee that there's gonna be no mutations, there's no natural selection, and populations large enough to eliminate random changes, random environmental changes. Individuals are random, that you already know it doesn't happen in humans, even some animals. And then no individuals are coming in, no migrations. No population really follows all these 5. So that's why it rarely happens for Hardy-Weinberg to be true. So allele frequencies are most of the time changing, but if they don't change, we follow the model called Hardy-Weinberg equilibrium. I just saw the chat. Okay, so 3 types of natural selection. So this is 3 types of natural selection. I want you to know definitions of directional, disruptive, and stabilizing. I will give you the definition and then ask you if this is directional, disruptive, stabilizing, or maybe just natural selection. All these are modes of natural selection. So it's directional when one of the phenotype is being favored. What is a phenotype? Do you guys remember definition of a phenotype?
The physical appearance.
Physical appearance.
Gene
So
characteristic.
gene characteristic that's expressed. So when the gene is expressed, whatever you have. So the directional, it's going in one direction. And one phenotype is favored over another. That would be the definition of it. Disruptive, extreme phenotypes are favored, and one intermediate phenotype is not favored. In this case, snails. And then last one was the stabilizing. So intermediate phenotype is being favored. So we talked about baby heads. If it's too small, maybe they're gonna have some cognitive issues. If it's too large, it's not gonna pass through the birth canal. So it has to be the right size. So that's gonna be favored over time. And then other ways, here's the slide. The evolution happens is, so one is natural selection, and it's not only happening through natural selection. Another one would be mutation and genetic drift, non-random mating, and migration. Migration means they're coming in. Non-random mating means they are in a sexual selection. So it's a type of natural selection. They are selecting for their mates. Genetic drift. Can somebody tell me what genetic drift is? I give you the example of pilgrims. So when a population is small, some of the alleles get selected, maybe in the first generation, because they increase in maybe numbers for the generation, they're gonna get selected more often. So here we have 50-50, we start with 50-50, but it's a small population. When you randomly select, 7 are orange, 3 are blue. So there's a higher chance more orange are gonna get selected from this stock. Now 70, now you select again, now 9 are orange. Because there are more orange, there's a higher chance they're gonna get selected for. 1 is blue. You restock it, now you have 90 orange, 10 blue. Now there's a very high chance only orange gonna get selected. And then over time, this allele disappeared. So that's genetic drift, that mostly happens in small populations. Then there was bottleneck and founder effect. Can you guys tell me what founder effect is?
Few individuals establish a new population and allele frequency might change.
Yeah, because they're only mating themselves among themselves. So they're only mating in that group. It's a small population, so the allele frequencies changes. So that's founder effect. And then there was a bottleneck. So that's when many members of a population die. Happened to the another population of I'm trying to think, I can't remember. In Big Sur, it happened in Big Sur to you guys remember which population I gave you for that? I know what they look like, I just can't think of the name.
Was it the sea otters?
Sea otters, yeah, sea otter population happened. And then there was a small population left in Big Sur. So they saved that population. They started, mating them themselves. Now there's a smaller, so most of them died. So there is a less allele diversity, I would say. So the diversity reduces and reduces the population size, and you had more alleles here when there was original population, and then here it went down. Only few alleles remain in the small population. So non-random mating. So mating is really random. That's known as sexual selection. And artificial selection, that's going to affect some of the allele frequencies, whatever gets them, maybe whatever attracts a mate, that allele is going to go higher in number, or that's going to be seen more often. That's going to be favored by natural selection. Then migration. So that's when some individual moves in or moves out. Actually, both are going to affect population. So migration moves alleles between population. It's going to affect this population because this individual is leaving from this population, and this population because it's coming in. So that's migration. This chapter was very straightforward. Any questions on this? No questions? I think 15 and 16 were a little bit more complicated. Okay, so I want you guys to define a fossil. If you did my play pause videos, I asked all these questions on there, and I asked you to define a fossil there as well. How do you define a fossil? How old does it need to be?
More than 10,000 years.
More than 10,000 years. What would you define as a fossil?
Preserved remains of an organism,
Remnants
an
of
ancient organism.
Ancient organisms. Okay. So what counts as a fossil? Here are some pictures. So impressions, I would say impressions count as fossil. What else?
Bones.
Bones.
Bones, organic matter, maybe plant resin. Maybe if something gets preserved in tar, maybe. So mud, stone. So all these count as fossils. I might ask you an example of a fossil. So that's why I'm letting you know that. Why fossils are missing links? Why are there missing links? So here's a perfect fossil. We have all the steps for modern horse from Heracitherium, but not all fossils are like this. So there are missing links. So that's why I said that some people say, why can't you show me all the evidence for it? So why are some fossils missing links?
Destruction due to environmental changes.
So destruction due to environmental changes. So if an organism is made up of maybe calcium carbonate shell, and then another one is made up of, I don't know, soft tissue, maybe a liver or pancreas, so that kind of tissue. So which one's gonna get preserved?
The shell.
The shell, right? So the shells that have hard structures, they get preserved more easily compared to like wings or soft bodied jellyfish. They're not gonna get preserved. So that's why we have some missing links, and also because we have the continents moving, and we have volcanoes, it's gonna destroy some of the fossils. So what does the fossils of Oklahoma that they found give you the evidence of? So if you find something like this in land, it's gonna tell you there was water there once. So that's the evidence the Oklahoma shells provide. So I want you to remember this. So I might ask you about this, Oklahoma. Okay, so this is absolute dating and radiometric dating. So absolute dating is a form of radiometric dating, or radiometric dating is a type of absolute dating. This is when you exactly date it. This is called radioactive dating. Do you guys know how to do this? There was a question on the study guide as well for this. If I say the half-life of this organism is 20,000 years, what percentage remains after 40,000 years? What percentage of the remains after 40,000 years?
25.
25%, because in 20,000 years, it's going to be 50% remaining, in another 20, it's 25% remaining. So there will definitely be a question where I give you some numbers and ask you about this. If you cannot do this, make sure you can do this. It's probably going to be worth more to this question. Can everybody do this? Can you do the absolute dating? Yeah, okay. This is an example of relative dating. So this is more recent compared to this, and then this is more recent compared to this. So if you go down in the layers of rocks, it gets older, but we don't have exact number. Then there is plate tectonics theory that I want you guys to remember. That everybody knows this. I don't think you have to remember this. So you guys already know that our surface is made of several layers, and these plates are moving. So you can see over time this will happen. So that's plate tectonics. And it impacts the populations depending on if they were together, there's more competition. Now there's less, now they can maybe, there's more variation over time. They diverge into different directions. So they create geographical barriers otherwise. This is Wallace's line. This is just a hypothetical line that's showing that there's different types of species found on this side compared to this side. Maybe the weather is different or the environment is different. So it gives you a variety of organisms on each side of the line. That's known as Wallace's line. Next important topic is homologous and analogous. So what is homologous?
It's the same.
Same structure.
The structures.
Yes, but where are these structures coming from?
Common ancestry.
And yeah, that's the word.
Common ancestry. For analogous, they have maybe similar structures, but it's not coming from common ancestry. It's coming from the selective environmental pressure, because the environment is same. Maybe it's causing all the fish to have the streamlined body shape. Which of these follows convergent evolution or example of convergent evolution? Is it analogy or homology? Which one is convergent evolution?
Analogy.
Analogy. Homology is divergent evolution. So homology, divergent evolution, analogy, convergent evolution. But before that, you have to know what is homology and what is analogy.
Professor, can you explain analogous structures one more time?
Yeah. So I'm gonna just pull up an example here. So analogous structures, an example of that would be this fish having the same shape as a mammal. And then the penguins also have the same body shape. And then this reptile also have the same body shape. All these have same body shape, but it's not coming, they don't have a same body shape because they have a same ancestor, or they have shared ancestry. They belong to different groups. It's a fish, it's a mammal, bird, reptile. But this body
So why do penguins eat fishes, Professor?
Why do they eat fish?
Yeah.
So let me finish this canal first. So let me finish explaining this. So they have the same body shape because they are living in the same environment, which is the ocean. So maybe that's the best body shape to have underwater. So the same body shape is coming from the selective environmental pressure because they're living in the same environment. It's not coming because they don't have that because they have the same ancestor. They belong to different groups. Does that make sense, Scott?
Absolutely. Yeah, thank you.
So Kunal, I don't know why they eat fish. It's their food source.
Thank you so much, Professor.
Mm-hmm. So divergent, and this is convergent evolution. Divergent is when we're saying they're sharing the ancestors. So here, so they have the same bone and it might differ a little bit, changes happen, modifications happen over generations, but this is, they're sharing common ancestor. So that's why they look similar, or they have the similar bones here. What are vestigial structures?
Structures there, the purpose is no longer utilized.
So they
Oh,
have
no, sorry. There's structures that have the same structure, like shape, but they have different functions.
no. So
I'm
you
close.
were
I
right
know
the
I'm close.
first time. You were right the first time, Chris.
Okay.
So vestigial structures, they don't have a function, but maybe they are functional in another species. So for example, snake legs, they may be functional in other species, but the snakes don't use it, but they still have those spots left. Whereas here, whale ancestor, they had limbs, but now they have these leftover structures. We have the appendix. We don't have a function for the appendix. Maybe in another animal, there's a function for the appendix. So it has lost its function, but it's homologous, similar to a functional structure in another species. So that's a vestigial structure. You can expect 5 to 6 questions on homologous and analogous. I will say you have a bat wing, you have a bird wing. Is this homologous or analogous? So go back and look at the examples, the ones I gave you. Also, some are in your book or you did in your homework. Then I might say you have a bat wing, you have insect wing. Now you already know they belong to different groups, and that's going to be analogous. So I might give you some questions like that. Then I might say in snakes, you have these I don't know, tiny legs or something left over. What type of structure is this? Homologous, analogous, vestigial? Then you're gonna say it's a vestigial structure because it doesn't serve a function. Or I might say these animals live in the caves. Now they don't have eyes, but one is a salamander, the other one is maybe belongs to another species. It's an arthropoda or something. I don't know. I think I gave you an example of that. So I'll tell you that. And then I'll say, is this homologous or analogous? If they belong to different groups, you're going to say analogous. So here, this is the example I gave you. Okay, and then the embryonic development. I want you guys to remember that the embryos in the embryo, human and chimpanzees, they look similar. As adults, they look different. So even other animals, so here's a chicken, mouse, and human, they look similar in the embryo, but as we grow up, we look different. And that's controlled by homeotic genes. Homeotic genes, if there's a mutation in those genes, it might cause some structures to grow incorrectly. So maybe some flies get heads on their, some flies get legs on their head, and that's maybe there's a mutation in homeotic genes. Homeotic genes are the ones that control development. So that's, if you're trying to remember what these are, you want to say they control the development. So organisms' development. These are very important genes. If there is a mutation in these, you can be limbless, or you can have limbs in a place where your head's supposed to be. So it's a very important genes. Now, then we can compare the DNA and see how related we are to chimpanzees, gorillas, orangutan. We can compare the gene sequences, so they can do that now. Then this last part is molecular clock. Now tell me, so answer this question for me. To estimate if the species are related using this molecular clock, what information do you need? What do you need to know if you have to answer the question how related these species are using the molecular clock? Maybe when were they?
Sequence.
DNA sequence?
The DNA sequence. What else?
RNA?
No, you are
The
looking at
base,
mutation.
I guess.
So you need to look at the mutation rate. So this is happening every 25 to 50, 25 million years. So there's one mutation, 50 years, another mutation. So you want to know how fast or quickly or when the mutations estimately is happening, every 5 years, every 10 years, every 20 years. That's one thing you need to know. Then you need to know number of mutations that differ between those species. So you can kind of go back and estimate that. And then it has to have the same gene in both species you're looking at. So you can't look at one gene and say there's a mutation in this gene. Then you look at another gene and you say there's a mutation in this gene. So those are some of the things you will need if you're trying to estimate the relatedness of two species or how long ago they were the same species. So you have to kind of think about it a little bit critically to see what you're gonna need for that. So I think that was the last question. And then sometimes it gets more simpler compared to complexity. So they have lost eyes, that's getting more simple. So the evolution doesn't lead to greater complexity always. Sometimes features are lost.
I have a question about that.
Yes, Eric?
Even though they don't waste energy, say for instance, creating eyes,
Mhm.
can't that energy be diverted to something that's more, advantageous?
Yeah, so maybe they do that. They don't want to put the energy to making eyes. Maybe they're putting the energy into sense of smell. So they can use the sense of smell to get more food because they can't see anyways in the dark. Yeah, so Chris said rate of allele change, rate of gene change, or mutation, how often it happens, names of change, number of change codons. So yeah, those are two already. And then the DNA sequence. So those are a few that you have to know. Any questions on this chapter? And last thing I want to say, these two species, they can't mate with each other now. They are different species. There's no gene exchange happening. They each have a different gene pool. So 14. So 14, this chapter, another name I gave you was Carolus of Linnaeus. Do you guys remember what he was responsible for? Carolus of Linnaeus?
Taxonomy.
So it's for naming things. He was able to name things, organize them. So his name is Carolus of Linnaeus. So I want you to know Charles Darwin. I want you to know Carolus of Linnaeus. What does the word speciation mean?
The beginning of a species.
So the word speciation means new species. So when you
An extinction is when the species doesn't exist anymore.
Yes, so extinction is when they die out or cease, so they don't exist anymore. What was the name of the first or the oldest fossil that I want you guys to know? Do you guys remember that? I forgot to ask you. That was stromatolite, and it was. Do you guys remember how long ago it was found?
Is it 3.8 or?
So 3.8 billion years ago. Another thing, make sure you guys know that mnemonic, kings play chess on fine green squares. So kingdom, phylum, order. So make sure you guys know that. In that whole system, the group that's most inclusive, means includes most of the species, would be the very top one, kingdom. Least inclusive is species because it's very specific. So make sure you guys know that. In this Homo sapiens, which one is a genus, which one is species?
Homo is genus, sapiens is species.
So Homo is genus, sapien is species. So make sure you guys can separate that. I might give you some examples and I'll ask you which one is a genus, which one is a species. And then very important, very, very important is biological species concept. You have to know the definition, which is their potential to interbreed and produce a fertile offspring. I might give you some examples. I might say you have a horse, you have a mule. No, so you have a horse and a donkey and they produce together and they produce a mule. Mule is infertile. It doesn't support biological species concept. Are they the same species or are they different species? In this case, they're going to be different species because they cannot produce a fertile offspring. So I might give you some examples like that. So make sure you guys really understand the biological species concept. Third name I need you guys to know, Ernest Mayr. So know Ernest Mayr. So he was the one who came up with biological species concept. Speciation, formation of new species. And then we started getting into reproductive barriers. You will get 7 to 8 questions on reproductive barriers. You have pre-zygotic barriers, you have post-zygotic barriers. Make sure you guys know which ones are which. I might give you name of a barrier. I'll ask you if this is a pre-zygotic barrier or if this is a post-zygotic barrier. Pre-zygotic is before you have the egg. So there is no mating that's happening. You don't get to the egg. That's basically before you have the egg. Post-zygotic, after you have the zygote, after you have the egg, it doesn't reach the adulthood stage. That's post-zygotic barrier. I gave you a list. There's a table I'm gonna show you, but make sure you guys know. So if you do Kahoot, I think I gave you 20 questions for this. So I might say these two species live on different environments. What type of isolation is this? What type of barrier is this? First thing you need to know is a pre-zygotic barrier. Second thing you need to know is a habitat isolation. Temporal, they are active or fertile at different times. So all these are pre-zygotic barriers, means some of these cricket species, they are fertile during spring. The other ones are fertile during summer, maybe. So at different times of year, therefore they cannot mate. That's a temporal isolation. Behavior, they might have different rituals. So frog makes a mating call. This frog makes a different mating call. They don't recognize each other's mating call. Therefore, they are not going to interbreed. Only the frog that recognizes this mating call is going to interbreed with this frog, even though they might be living in the same space. Mechanical, think of the orchid and the wasp, or the actually better example is the bat and that flower. So if the bat didn't have the long tongue, it wouldn't be able to get the nectar from the flower. So that's like coevolution. So mechanical is when the parts don't fit together. If it didn't have the long tongue, it wouldn't be able to get the nectar or pollinate that flower. So that would be a mechanical isolation. Gametic, sometimes there are chemicals that are found on the egg and the sperm. So these coral, you can think of sea urchins actually, they release their eggs and sperm in water and hope they'll find each other. But sometimes if there are these chemical receptors that the sperm doesn't recognize on the egg, or they don't, kind of fit together or work together, then it won't be able to fertilize this egg. So the gametes cannot unite because of some of the chemicals. So that's gametic. So you have 5 types of pre-zygotic reproductive barriers, or pre-zygotic reproductive isolation. And these are the barriers. Usually I see students having a hard time with these ones, the post-zygotic ones. Post-zygotic, you need to understand the fitness of the adult is reduced. Therefore, they're not going to be able to reach adulthood. If they do reach adulthood, they might not be able to reproduce. They're infertile. If they do reproduce, their offspring has reduced fitness. Therefore, it's not going to be in good health. There's going to be a breakdown of health. So let's look at some examples. Inviability, hybrid inviability. It doesn't reach adulthood. Something happens, it fails to reach maturity. So this plant species, eucalyptus, and that's for a hybrid. That means you already have a zygote of a hybrid from 2 different species, and now it's going to be able to see. So the nature is preventing basically forming hybrids through all these different things. Hybrid infertility. So this would be horse and donkey reproducing together. Now they have a mule, or a lion and a tiger. You have a liger, and this liger and the mule, they cannot reproduce. They're infertile. It's a post-zygotic barrier. Therefore, they spent all that energy making a hybrid, but the hybrid cannot even pass on their genes. Hybrid breakdown. This is when the reduced fitness happens. This means the hybrid is able to become an adult and reproduce, but the offspring has a reduced fitness. In this case, these mosquitoes have abnormal genitalia. That's hybrid breakdown. There's some issues with the body. So these 3 are post-zygotic. So First thing, you will have to be able to tell me which 3 barriers are post-zygotic. I might give you a list, you have to pick the ones that are post-zygotic. Then you have to know what each one of these means. And I might give you an example. I might say you have a lion and a tiger, you make a liger, which post-zygotic barrier applies here. Then you're gonna say hybrid infertility. I'll tell you that if they're able to reproduce or not. So those are some important ones. Make flashcards on these. Then we talk about the spatial patterns of two types of speciation. We have sympatric speciation and allopatric speciation. Please do not confuse speciation with the reproductive isolation. That's different. That might happen because, you know, there's a physical barrier, which means that's allopatric speciation, but do not confuse that with the speciation, the word speciation. In allopatric speciation, there's a physical barrier in between. Therefore, the genes from here cannot get here, so there's no exchange. They have a different gene pool here, different gene pool here. So that's allopatric speciation. There's a physical barrier, keyword, physical barrier. Sympatric, they're living in the same space. There's no physical barrier, but they still don't reproduce together. So populations diverge while living in the same space. You guys remember that cichlid fish? Maybe because they feed on different food sources. This fish feeds on here on shallow water, and the other fish feeds on deep water. They are living in the same space, but they still don't reproduce together. Maybe they can, maybe they're able to, but they just don't. So those are, then over time, they're gonna become completely different species. They're gonna diverge in different directions, and they won't be able to do the gene exchange because they won't be able to reproduce. That happened to those crab species or something, I can't remember. Okay, and then we have polyploidy. Polyploidy. Oh, one last thing I want to mention about allopatric and sympatric. Sympatric can have different food source, like the cichlid fish, and then that's going to cause the species to diverge. And then in allopatric, the physical barrier causes the species to diverge. What is polyploidy? Is this type of sympatric speciation or allopatric speciation?
Sympatric.
Sympatric. So polyploidy is when you have multiple number of chromosomes. If I ask the definition, you're gonna say that. And then if I ask you if this is a sympatric or allopatric, you're gonna say sympatric. And then it happens a lot in plant species. So we've been talking about ferns in the plant chapters. It happens in ferns a lot. Almost 95% of the fern species. And then it's increased chromosome number. And it happens naturally. And now in this cotton plant, you have two types of plants, and then they produce a hybrid, and then somehow the chromosome numbers naturally doubles. Now this plant cannot go back and reproduce with this parent plant. So it happens naturally in 50% of all the plant species, but it happens a lot in fern species, fern plants. That's polyploidy. Then we have gradualism. So can you guys tell me the difference between gradualism and punctuated equilibrium?
Gradualism is.
Gradualism.
Go
Go ahead, Justin.
No, it's all right. Go ahead, man.
Okay. Gradualism is a change in evolution that is slow and really able to be seen, whereas punctuated kind of happens all at once. And then I think it's because typically it's an environmental change that causes the species to have to change.
Yeah, so gradualism, change happens in small incremental, small increments. That's how the change happens over time. Punctuated equilibrium, it's a stasis. So stable for a long time, then there's a burst. Then it's going to change all of a sudden. So this flower, stable for a long time, then it changes all of a sudden. So long periods of little change or no change, and then a rapid change. That's punctuated equilibrium. So know the definitions for both. Ocean species, they are, so they're in the stable environment. So mostly that's gradualism. Maybe they're changing a little bit over time. The environment is pretty stable. Then we have the pace of speciation. So when there is a mass extinction, it's gonna open up a lot of new resources for the species that survived the extinction, basically, and then they can get more diverse. So when the dinosaurs existed, mammal species were still around, but they were really tiny. Then volcanoes happened, meteorite hit, and competition happened. That all the dinosaurs went extinct. Now that opened up space for these mammal species, and they got more diverse. So it can open up new resources for the surviving species of the extinction. So the evolution might happen pretty quickly when there's a mass extinction. So following a mass extinction. Species go extinct when all of the members have died. So when they all die, it goes extinct. Adaptive radiation is few species turn into many different species. So we have two types of extinctions. We have a background extinction. So we have some species, 1 or 2 species every year going extinct. That's background extinction. And then we have mass extinction. That's when lots of species die. So during this Carboniferous period, lots of moss species and seedless vascular plant species, they got burned down. And that's, I keep saying, that's where the petroleum and coal is coming from, this mass extinction. Background is 1 or 2 species go extinct due to gradual environmental change. So environmental changing gradually, now not gradually, I would say. Humans are changing it pretty quickly. And panda species and condor species, they would have gone extinct if we wouldn't have kept them, because maybe they weren't able to compete for food anymore, but human interference helped them survive longer. What is impact theory? Actually, before you answer that, I have another question. What causes mass extinctions?
Natural disaster.
An event, yeah.
So natural event would be maybe collision between Earth and a meteorite, because that's going to create a cloud of dust around. Sunlight cannot get to plants anymore. If plants going to die, animals can't eat the plants. So it's going to cause a mass extinction. Another one might be the shifting of the continent plates. That can also cause it.
The volcano?
Volcano, yeah. Shifting of the continent plates can lead to a volcano that can cause mass extinction. So the impact theory says that meteorites or comets caused some mass extinctions. The way they know, there is a chemical called radium. It's not found on Earth that much, but it's found in the meteorites. So when they find these bones of dead animals, they find the layer of that chemical on there. They know this happened, and they're not going to find like few bones. They're going to find lots of bones. Then they know this happened because something hit the Earth, and that's why we had the chemical on there. And then we are causing the 6th mass extinction. Maybe get yourself a Tesla, so you're not creating a lot of pollution. So we're causing the 6th mass extinction, and it's leading to habitat loss, fragmentation, creating pollution. We are also introducing non-native species, and we are over-harvesting, for example, hunting. So all this is leading to extinction for some species. This is what I said in the beginning. No difference between taxonomy and phylogeny. You create a phylogenetic tree when you want to see the relatedness between species. Phylogenetics is a study of evolutionary relationships among species. Taxonomy is the science of describing, naming, and organizing them or classifying them. So this is your mnemonic. Know this. I'm not going to ask you to know this. I'm asking you about genus and species. Know that genus is written before species. Allo is genus, Vera is species. And then know the order. If you know this mnemonic, you already know the order. Domain is not here. So domain comes first, then you can apply the mnemonic. Then definition of a clade, cladogram. This is a cladogram. So when you create the phylogenetic tree, it's called a cladogram. And that one clade is a group of organisms that share common ancestor. This is one clade, this is one clade, they're sharing common ancestor. Birds are more related to dinosaurs than they are to crocodiles. So this is a phylogenetic tree.